Well, dear students of 12th class, today in your botany we will try to learn about the concept of dominance. As you must be remembering that in the previous class we tried to explain the incomplete dominance, wherein an example quoted from the by Antirrhinum species or the snapdragon, or most commonly known as the dog flower. It was quoted, and when the inheritance in this plant was studied, the plant is having the two types of flowers, one with red color and the other with a white color. The red one obviously is the dominant one, and the white, that is the recessive one, represented here by the small letters, red by the capital R, and the white by the small r. And when the gametes are produced, there will be only the half of the chromosomes, that is the number of these chromosomes is reduced to half due to the meiosis, and only one of one out of the two allelic pair will be passed on to the next generation, so that the two get paired from either species for a particular character. And here what happened in this incomplete dominance, that the red colored flower gene was not dominant over the white one, not completely dominant, and it was partially dominant. And due to the this was the reason that in the F1 generation all the plants were produced with pink colored flowers, and in the F2 generation we had a ratio of 1:2:1 again, with one pure red, two pink, and one pure white, and this was due to the segregation. And now today we will try to understand the concept of dominance. What exactly the dominance is? Why are some alleles dominant and some recessive? And to tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. Every gene contains the information to express a particular trait. In diploid organisms, there are two copies of each gene, that is, as a pair of alleles. And now these two alleles need not always be identical. And here comes the dominance and the recessiveness into play. Now these two alleles need not always be identical. As in a heterozygote, one of them may be different due to some changes that it has undergone, and which modifies the information that a particular allele contains. It may have got modified in one way or the other, and the other will be the normal one. And let us take an example of a gene that contains the information for producing an enzyme. A gene that contains the information for producing an enzyme. Now there are two copies of this gene, the two allelic forms. And you must remember this, that there is a gene that contains the information for producing an enzyme, and there are two copies of this gene, the two allelic forms. And let us assume that the normal allele produces the normal enzyme that is needed for the transformation of the substrate. And theoretically, the modified allele could be the second one, that is the modified allele. The modified allele could be responsible for production of first the normal or the less efficient enzyme, number two a non-functional enzyme, or number three no enzyme at all. Remember one thing, that there are a pair of genes, that is the allelic pair, responsible for producing a particular enzyme, and one of the gene will, out of the two the genes, one will function normally, and that is most probably the dominant one, and other is going to act differently, that has got modified due to one reason or the other, about which we will deal in the later classes. And here this modified allele may be responsible for the production of the normal or less efficient enzyme, a non-functional enzyme, or no enzyme at all. And now, in the first case, the modified allele is equivalent to the unmodified allele. It will produce the same phenotypic trait, that is the result in the transformation of the substrate. Here we are studying the effect of this gene on the production of this enzyme. And such equivalent allele pairs are very common, but if the allele produces non-functional enzyme or no enzyme, the phenotype gets affected. It is to be affected. The phenotype, and this is to remember here, that the phenotype got affected because of the modified gene. The phenotype trait will only be dependent on the functioning of the Now, the phenotypic trait will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represents the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. And in the example above, the recessive trait is seen due to non-functional enzyme, or because of no enzyme is produced by the gene that got modified. Hope you understand, and thanks. Have a nice day.